friends, welcome back to this video, and we are going to today study about the basics of the enzymes. I'm going to discuss very important facts that everyone who is dealing with the enzyme and needs to study enzymology need to know about it. Okay, so stay tuned to the video. So the Edward Buchner, he was the person who coined the term enzymes. Okay, so the literal meaning of enzyme is, okay, the enzyme meaning, the word enzyme means yeast. Okay, in yeast. Okay, so basically all enzymes, they are protein. Okay, they are protein. However, there are some RNA molecules, okay, that they are been found to act as a biocatalyst. Okay, and those are known as ribozymes. Okay, they consist of one or the more polypeptides that are held together. Okay, and which bond is helping for these keeping this polypeptide chain together, the non-covalent bonds. Okay, so basically enzymes, they are the biocatalyst. Okay, so enzymes are biocatalyst. Okay, and these will catalyze various reaction like intracellular or extracellular. Intra means inside the cell, or extracellular means outside the cell. Okay, extracellular biochemical reaction. Okay, which kind of reaction the enzyme is uh, catalyzing? It is catalyzing the biochemical reaction. Okay, so the very fascinating thing about the enzyme is that they are like very effective. Okay, even if they are present in very traces, also they are very effective in their action. However, they are unstable. Okay, and because they are unstable, they can be easily inactivated or denatured. Okay, and this could be achieved by like temperature. So they, we can, in other words, we can say that they are unstable and they are easily influenced by the factors like temperature and the alkaline and acidic condition. Okay, that is the pH. Okay, now the enzyme, okay, they will act on the substances. Okay, so these enzyme, they will act on substances. Okay, those substances where the enzyme will act, they are known as substrate. Okay, okay student, and upon their acting, the substances will get converted into new substance, and that substance is known as product. Okay, that is produced after the biochemical reaction. Okay, so I will repeat it again, student, here carefully. The enzymes, they are the protein. Okay, they can act in the intercellular and extracellular biochemical reaction. They will act upon a certain kind of substances. Those substances are the known as substrate, and once the enzyme catalyzed reaction, it will lead to production of certain substances, and those substances, they are known as products. Okay, so let's talk now about the enzymology. So from the name only, it is very clear. Okay, logy means study, and enzyme. Okay, so the study that is dealing with the study of enzyme is known as enzymology. Okay, now let's quickly talk about the structure and the composition of enzyme. Okay, because in order to understand the enzyme, we need to know its structure, what it is composed of. Then only we will understand, right, how it catalyzes the reaction. So basically the enzymes, okay, made up of the proteins, okay. So there are some simple enzymes that are completely made up of protein, and those, okay, protein made enzymes, they are called as simple enzyme. Example here is the amylase. It's a simple enzyme. It is wholly made of protein, okay. But most of the enzymes, okay, they are made up of two portion, okay. The first portion, most enzymes, they have two portions, okay. The first portion is the protein part, okay, that is essential. And in addition to the protein part, they will also have a non-protein part, okay. So the enzyme having these protein part and the non-protein parts, they are known as conjugated enzymes. Conjugated enzymes. Okay student, it's that clear to you? The enzyme that is made up of protein, they are known as simple enzymes, okay. And the enzymes that are having the two part, that is one is the protein part and another is the non-protein part, they are known as conjugated enzymes, okay. Now the major portion of the enzyme, conjugated enzyme, is the protein part, okay. So that protein part is known as apozyme, okay. So what is apozyme? The major portion of the enzyme is the protein part, and that protein part is known as apozyme, okay, or apoenzyme also. Now the proteins part, okay, is in the conjugated enzymes, they are supplemented with the non-protein part also, okay. Now this non-protein part is also essential for the enzymatic activity, okay. This non-protein part, they, when they tightly bonds to the protein part only, it is called as prosthetic group, okay. So when this non-protein part, it is attached to the apozyme, that is the protein part, then they are known as prosthetic group. And when they are not attached, they are known as coenzyme or cofactor, okay. Okay, so I will explain you again. Most of the enzyme, they are made up of two parts, that is the protein part and the non-protein part. The enzymes containing the protein part and the non-protein parts, they are known as conjugated enzymes, okay. When this non-protein part is attached to the protein part is known as apozyme, okay. So when the non-protein part, it is attached to the apozyme, then it is known as prosthetic group. And if it is not attached, then it is known as coenzyme or the cofactor, okay. Then the question arises in our mind, what is the complete enzyme? So the complete enzyme is defined as enzyme that will include the prosthetic group, okay. Then the enzyme is known as complete enzyme, or it is also known as holo enzyme, okay. So I will draw it for you. Suppose this blue is the enzyme, okay, protein part, apozyme, this is apozyme, okay. Now it has a space for the coenzyme or the factor, okay. Okay, now this coenzyme, when it will sit here, okay, and this is the active site, okay. This site, this cavity is the active site. I will write it for you. This is the active site. Now the holo enzyme or the complete enzyme, it will have the protein part, that is the apozyme having the activate site. Okay, and this prosthetic group, it will just sit on here. Okay, it will fill this. Then only when it is tightly fitted or attached, then only it is known as holozyme. So without the prosthetic group, the enzyme protein part is known as only the apoenzyme only. Okay, but when apoenzyme is attached with the prosthetic group, when they are together, okay, held together, then it is a complete enzyme and it is known as holoenzyme.